Susy has 30 years of experience in research and development in human and pet food industry, and she was recently retired in March this year. She's the founder and president of TPS Consulting. In her last position as Chief Scientific Officer at Instinct Pet Food, she has developed a science-based approach in making safe raw pet foods. I hope that the young man that was asking about safe pet foods will have the time to be back after lunch to hear this talk. While at Instinct, she collaborated with outside institutions such as the University of Nebraska-Lincoln, University of Arkansas, Log 10, and Institute of Food Safety and Health, Illinois Institute of Technology. And in 2018, she initiated a multi-year research collaboration with the University of Helsinki and us, the DogRisk Group, to build a scientific body of knowledge for the health benefits of feeding raw to pets. And it's the first time actually that Susy is on having a talk for us, and we've been looking forward to this talk for many, many years. And so I give it to you, Susy. Thank you for coming, and please, you may start your presentation.
Thank you, Anna, and hi everyone. Hopefully you have had good lunch, and if you cannot hear me very well, maybe you can also type or let us know. But we checked my voice before, and Anna said that you guys can see or can hear me very well. So before I start, I would like to acknowledge my previous company. As Anna said, I work for Instinct Pet Food, who provided funds for the validation studies conducted at the University of Nebraska in Lincoln and the Department of Food Science and Technology and the Illinois Institute of Technology, Institute for Food Safety and Health. And I would like also to thank you, those people whose name I listed here. So this is a slide to show the various forms of pet food sold in the market here in the US. The first two on the left are the traditional ones, retort or canned and extruded or dry kibble. And to the right of the dry kibble shown here are all the alternative pet foods, like from baked to air dried, all the way to frozen and freeze dried. And they are becoming more and more popular. People are looking for these alternative foods because research has shown that foods that have high heat treatment, such as the one used to make canned and dry food, generates a high amount of AGEs. And you've heard from Dr. Wynn before that, yes, we have had research to show that the amount of AGEs in canned and dry kibble is very high compared to raw frozen. And there are research already, as Dr. Wynn already shared with you, that this may negatively affect the pet's health. Of course, today my topic will be focusing on the use of high pressure processing, or in short, HPP, to make raw pet food. Excuse me. Before I start talking about HPP, I want to point out in the USA about the regulatory paradox. The FDA has mandated all pet foods to contain 0 pathogens. Raw materials that comes from USDA inspected facilities, like poultry and meats used in pet food, may contain pathogens, as the USDA allows the presence of pathogens excuse me, in poultry and red meats. As soon as the raw materials come from the USDA inspected facilities are received on the pet food dock, the FDA oversight kicks in. AFCO, which is the equivalent to FEDIAF in Europe, does not have oversight for pet food. It doesn't provide any guidelines on poultry and red meats for pathogens tolerance. AFCO only provide guidelines on ingredient definition, nutrient profiles, and labeling. Excuse me. This slide shows the standard performance of the USDA for human-grade poultry, pork, and beef. I'm not going to read through all the numbers here. I just circled 4 of them to give you a flavor on the percentage of allowed positive salmonella on human-grade poultry and meats here in the US. You can see here that all human-grade poultry and meats do contain pathogens, as they are meant to be cooked before consumed. This is important because some raw pet food companies using human-grade poultry and meats give the impression that their meats or poultry do not contain pathogens because they are of higher quality. And consumers do not know that using human-grade poultry and meats does contain pathogens. So their assumptions are that they are free of pathogens. Are the poultry and meats the only source of risk of pathogens in making safe raw food? Well, there are multiple areas that can be a source of cross-contamination for pathogens while making raw pet food. Let's take a look at it. So this process flow chart is a typical one for raw pet food company that use HPP as a non-thermal little step. Thus, in this case, the process can be broken down into pre-lethality steps and post-lethality steps. The process starts with raw materials, meats, dry ingredients, and liquids being received. The meats are then stored in a freezer while the rest of the ingredients are stored in either regular room or cold room. Before the frozen meat are used for production, they need to be thawed out first to reach the desired temperature prior to grinding. All ingredients are weighed and then blend together and form into chubs weighing between about 5 to 15 pounds. And the next step is the HPP step, and anything after HPP, like I said before, is considered as post-lethality steps. After the chubs are HPP, they are then ground, form into its final form, whether it's bite-sized, patties, nuggets, or other forms. Once they are formed, then they are frozen, packed into their final packets, and then sent to the freezer before being distributed. So after showing you all these steps of making raw pet foods, where do you think pathogens could potentially cross-contaminate the food? Well, here's the answer to that questions. All of them, except when the product is already in the final package and kept in storage. And that's assuming that the package is not leaking or there's no pinholes. So how do you keep raw pet food raw and make it safe and still maintain the nutrient content of the ingredients? So let's talk about HPP now. In the next few slides, I will show you how it works and talk about fundamentals of HPP. For some of you, the information I'll be sharing here may not be new, so just bear with me now. What is HPP? HPP is a non-thermal food preservation technology that uses water, and more than likely it's chilled water and high pressure to inactivate foodborne pathogens and spoiled microorganisms. And where is HPP used? In human food, HPP is widely used to make salsa, avocado, deli meats, seafood products like raw oyster, and some ready-to-eat meals like soups, pasta, potato salads, and dairy products like yogurt or smoothies. HPP is also used in pharma and cosmetic industry, and last but not least, in pet food. Now, how does HPP work? First, airtight airtight sealed packages are loaded into carrier basket. You can see in this picture here, the cylinder tubes are in sealed packages inside the basket. The baskets are then inserted into the HPP vessel, which is this one here, and they're inserted here, which is then sealed, or the chamber is being closed and pressured. The product is held under pressure for a few minutes, called the dwell time, depending on the recipe. As I said before, HPP is a non-thermal process. It inactivates harmful bacteria and food spoilage organisms. It does also kill the good bacteria, contrary to some raw pet food companies' claim. It does not break covalent bonds. So small chemical like flavor, vitamins, minerals are not destroyed. And it retains most heat-sensitive bioactive compounds such as polyphenols and antioxidants. It only affects hydrogen and hydrophobic bonds, which are non-covalent bonds. So in the next 3 slides, I'm going to try to explain why small compounds like vitamins and minerals are not affected by HPP. I'm using the subway technology here, subway analogy here. So I'm using happy face to represent a small compound such as vitamin or mineral. And the water droplets here are representing the water in the food that surrounds the vitamin or mineral. Just like in a subway, when there is not many people around you, and you just jump into the subway in the first station when it is not during peak hour, you're free to move around, and you may be even able to stretch your leg or extend your arms. Imagine this scenario as a small compound like vitamins and minerals. They are surrounded by the water in the food, and they're relatively free to move around within its surrounding. So when more people jump into the subway as it goes through more stations, you start to feel a little bit squeezed and not able to move around in a crowded subway. Using this scenario, when the pressure in HPP is raised, it squeezes the water in the food, and the water gets closer to the small compound. But the small compound is not free to move around, but still remains safe. So when you are in the last station of the subway, more likely there is less people because a lot of people have left in prior stations, and you are free to move around again. In the case of HPP, when the pressure is released at the end of the cycle, the small compound is back to its original state. So this is a slide to explain the 4 structures of protein, from primary, secondary, tertiary, and quaternary structure. I'll let you read quickly the various types of protein structure yourself. But what's important here that I want you to focus on is the difference in the sensitivity to pressure of these 4 structures. The quaternary structure has the highest sensitivity to pressure because the amino acids have weaker interaction with fewer bonds and chemical interactions among each other, while the primary structure has lowest sensitivity to pressure because the amino acids are connected via hydrogen bond. And the hydrogen bond is a covalent bond that's, it's connected via hydrogen bonds, but they are very strong bonds. So what's the significance of this? This means that HPP may alter the space arrangement of quaternary, tertiary, and secondary structure of protein, but not the primary structure. In other words, the nutritional value of a protein remains unchanged with HPP. So with the explanation I just gave you, it should not be a surprise to you that the color change of meats may change with HPP because the color change is associated with changes in the spatial arrangement of myoglobin, which give meat its red color. Here you see 4 different meats: beef, pork, turkey, and chicken. And on the top, it shows the pressure scale in megapascal from very low to 400. In beef, you can see the gradual color change as the HPP pressure is raised. Color change starts around 150 megapascal and becomes more apparent above 250 to 300 megapascal. In other meats like pork, turkey, and chicken, less color change is observed and HPP pressure gets higher since as HPP pressure gets higher since these meats have less myoglobin in them. Let's discuss the effect of HPP on various microorganisms now, since the purpose of using HPP in raw pet food is to kill pathogens. HPP is unique in that it applies pressure uniformly throughout a food, regardless of size, shape, and geometry. Let's talk about the amount of pressure used in HPP. You probably know that the Titanic wreckage lies at the bottom of the sea about 700 kilometers southeast of the coast of Newfoundland, which is the most easterly province in Canada. It is about 4 kilometers deep, and the pressure at 4 kilometers deep is about 40 megapascal. In raw pet food, the HPP pressure used is normally around 580 megapascal or 85,000 psi. This is 14 and a half times higher in pressure than the pressure experienced at 4 kilometers deep. So what happens to the microorganism cell at this very high pressure? The mechanism is believed to be through disruption of cell membrane. The term we coined is death by leakage. Here, it's shown high-resolution photos of 3 different pathogens: E. coli, L. mono, and Salmonella enterica. These upper photos show a cell without HPP or in its original shape, and the lower photos show the shape of cell that has been HPP. As you can see here, there are cellular changes of the cells after they are being HPP. And this slide illustrates the cellular changes with E. coli, but what happened in E. coli, the same thing changes also happen with other microorganisms. On the top, we have healthy cells, and at the bottom, we have injured cells. Some of the effects of HPP are inhibition of protein synthesis through dissociation of ribosomes and inhibition of DNA synthesis, promotion of oxidative stress, and denaturation of intracellular proteins which affect metabolism. So what microorganisms are inactivated by HPP? HPP inactivates vegetative bacteria like Salmonella, Listeria, E. coli, O15787, Aztec, Campylobacter, viruses, yeast, and molds. So this slide shows how HPP affects different organisms based on their complexity. The most pressure sensitive is the most complex group. They are vertebrates like fish, amphibians, reptiles, birds, mammals. Invertebrates like sponges, jellies, animals, worms, and arthropods. And plants, they're all killed below or inactivated below 50 megapascal. Next is the group of parasites. We are still rather complex, but not as complex as vertebrates. Then molds and yeast, which is higher, or it's more resistant to pressure, but it's still lower than virus. And the viruses are started to get inactivated between, I would say, 200 to about 350 megapascal. And the next group is bacteria, mainly the vegetative bacteria. The vegetative bacteria started to get destroyed or inactivated around, I would say, about 300 megapascal all the way to 500, above 550, say about 600 megapascal. And Listeria, being a gram-positive, is more resistant to high pressure processing than Salmonella or E. coli. Bacterial spores, on the other hand, cannot be inactivated by HPP because the amount of pressure needed to inactivate spores is above 600 megapascal. And to inactivate spores, you may also need a combination of HPP and heat treatment. So that's one of the, I would say, the weaknesses of HPP is not being able to inactivate bacterial spores. So the performance of HPP is affected by processing parameters and product parameters. As far as process parameters, it has been shown that pressure and dwell time are the two most critical processing parameters affecting HPP performance. For product parameters, there are a few things that have been shown to affect HPP performance, such as pH and water activity. On the left here, the effect of pH on Listeria, E. coli 015787, and Salmonella enterica is shown. The pH started at 6.5 and 5.5 and 4.5. So the lower the pH, the higher the inactivation level or the log reduction. So the higher the bar, the higher the inactivation level. So it clearly shows that the lower the pH, the better the HPP performance is. On the right hand here, it shows the effect of water activity on the activation of in this specific graph is just the Listeria monocytogenes. The highest inactivation level is at water activity of 0.97, and the lowest inactivation level is at 0.86. Thus, HPP again is most effective at lower pH and higher moisture. So if HPP works, the question you might ask is, why do we still hear raw pet food being recalled due to the presence of pathogens after they're being HPP? Well, the key here is that the manufacturer needs to do validation studies to understand the variables and their impact on HPP performance. This is not a comprehensive list, but here's a list of some of the variables that need to be considered as part of a design of experiment to validate the effectiveness of HPP in killing pathogens. I've briefly mentioned about the importance of HPP pressure and dwell time. Choosing the right pathogen cocktails to include, for example, do you include E. coli aztec or not? If yes, which strains of its pathogens in the cocktail should be included? Because choosing the right pathogens cocktail and which strains to include is one of the key discussion that the manufacturer is advised to discuss with the representative from the FDA. One of the things that you also have to consider is the level of inoculation of the cocktails. You want to inoculate at a very high level, because if you inoculate at a low level, like 2 to 3 logs, then the results will show that HPP kills all of them. So you really need to challenge the HPP performance when you're designing the validation study. In regards to what proteins to include, you need to include also not only just the chicken, because different proteins may behave differently to HPP treatment. And it has been shown also the formulation may have an effect on the HPP performance between low-fat formula versus high-fat formula behave differently when it's being HPP. Then there are also operational factors to take into account, like grind size and tempering temperature prior to HPP that needs to be factored in. Tempering or increasing the product temperature prior to HPP up to a level may be necessary, as low temperature possibly mask ice crystal and impact the HPP performance. What about storage temperature for HPP? Will refrigerated versus frozen storage temperature potentially impact injured cells to recover? And speaking about injured cells recovery, one should also ask, should the product be tested, the product after HPP, should it be tested for pathogens immediately or 24-hour or 48 hours post HPP to take into account potential recovery of injured cells or injured cells that may die off, that may not die off right away? If the product is tested immediately, it may give a false negative or positive. Lastly, one of the most common questions is the effect of HPP on nutrients. Are there any nutrients destroyed? If yes, which ones? Remember, in previous slides, I have shown you that bigger compounds and weaker bonds are affected by HPP more than smaller compounds and that have stronger bonds. So I mentioned earlier that tempering temperature of the product prior to HPP is important, as it impacts the effectiveness of HPP. On this graph, the temperature is shown on the x-axis and pathogen log reduction is shown on the y-axis. Salmonella log reduction is shown as solid blue rectangular, and Listeria mono is shown as just regular blue rectangular. At 0 degrees Celsius, the log reduction of both Salmonella and L. mono is only around 1 to 2 logs. But when the product is tempered and the temperature is slightly raised to above 0 degrees, the ice crystal starts to melt. And as the temperature is raised to above, say, around 36 to 39 degrees Fahrenheit or 2 to 4 degrees Celsius, more ice crystal disappear. Salmonella log reductions improve from less than 2 to 4 or 6 logs, while Listeria mono went from around 2 logs reductions all the way to greater than 5 logs reductions. Thus, optimum product temperature prior to HPP appears to be above freezing to get the highest log reduction. There are two things to show here, HPP dwell time and the agar media used to enumerate the microorganism after HPP, after the product is HPP. The gray bars are product that either not HPP or the control or HPP at 3 to 6 minutes, and the microorganisms are then plated in a TSA agar. The blue bars are for product that either not HPP or HPP at 3 minutes or 6 minutes. Then the microorganisms are plated in a selective agar media for Salmonella. The longer the dwell time, the larger the log reduction, as you can see here. You can also see here that when using a non-selective media like the TSA agar media, the log reduction doesn't seem to appear to be as good as when selective media, which is the blue bars here, is used. But when you're using the non-selective media, the bacterial count may not be as accurate because Bacteria other than Salmonella may grow and be counted as Salmonella. So the key messages here is selective media needs to be used for each specific pathogens of target to get the true picture of how many log reduction is achieved. This slide shows the impact on taking product samples immediately after HPP at 0 hours, 24 hours, 48 hours, or 72 hours later. The first graph here, the blue bar here is the control or not HPP. So it's no surprise that the number of pathogens, remember that this is a challenge studies, so the number of pathogens is very high, as the listeria, in this case listeria, is inoculated around 7 logs in this experiment. The other 4 blue bars on the right here are all products that have been HPP, and samples were taken either at 0, 24, 48, or 72 hours after each HPP, and enumeration was done. Immediately after HPP or at 0 hours, less than 200 cells were recovered, and the number of recovered cells are up in 24-hour sample, as some injured cell may not die off completely. This trend seems to be true also for the 48 hours and 72-hour samples. The key messages here is to know, for the manufacturer to know when is the best time to pull samples before the product is released. You don't want to do it too soon or too late to avoid a false negative or positive. So as I mentioned earlier, one of the variables that may impact injured cells, whether they are eventually die off or recover, is storage temperature. This slide shows microbiological results of chicken formulas stored at 4 degrees Celsius over time, from 0 or 1 day to 21 days for Salmonella, E. coli, and Almani, after the product is HPP at 586 megapascals for 4 minutes. On the y-axis here is the log reduction. The 3 different colors denote different formulations. They're all chicken-based, but the composition of each formula is different. In the case of Salmonella, greater than 5 log reduction is achieved regardless of formulas over the entire storage time. In the case of E. coli Aztec, greater than 5 log reduction is achieved regardless of formula over the entire storage time. Specific to the R formula, which is in yellow bars here, on day 1, there is 5 log reduction, but on day 21, the log reduction is about 6 log. In the case of El Mano, it behaves differently than the other 2 pathogens, because El Mano is gram-positive and more resistant to HPP than Salmonella and E. coli. The log reduction on El Mano on day 1 varies quite a bit depending on the formulations, and it varies from 2 to 4 logs. But the log reduction gets better on day 4 and day 6, but drops on day 8 on all formula and increase again on day 13 on all formula and drop again on day 21. This inconsistency is rather interesting, and it is we speculated that some injured cells recover during storage over time, as El Mano is known to do well in refrigerated temperature. And El Mano, like I said, is more resistant to HPP treatment than Salmonella and E. coli. This slide shows the data of log reduction over time when the HPP product is stored in frozen storage. All other things are the same except for the storage conditions. Let's just focus on the El Mono, since it's the pathogens of interest from the last slide. In frozen storage, it appears that the injured cells are not able to recover as much as we've seen in the refrigerated storage, even though some variability is seen from one day to another, but it appears that the error bars are not as big compared to those seen in refrigerated storage. Here's the data to show the effect of formulation matrix and protein type on HPP performance. Beef formulas are on the top left-hand side, chicken formulas on the bottom left-hand side, and lamb formulas in the middle. For beef and chicken, there are 3 different formulas, and they're all different in composition. While lamb, there are only 2 different formulas. All data are from frozen storage for El Mano. The x-axis shows days of storage, and the y-axis shows log reduction. For beef and chicken, the A formula, which is shown in red bars, consistently achieve a higher log reduction than the S formulas, which is shown in green bars here. Well, the lamb formula is the other way around. On lamb formula, actually, the S formula has higher log reduction than the S formula. For beef and chicken, the R formula, which are shown in blue bars here, are consistently which achieve consistently less than 4 log reduction and lower than the other 2 formulas. And the reason for this is because the R formulas have the lowest water activity or lowest moisture content. You recall in previous slide that water activity of the formula does affect HPP performance. Last but not least, this data was collected to answer the question on which nutrients, if any, are destroyed by HPP. This is the most common questions that consumers, veterinarians or store owners ask. In this study, all nutrients that have an AFCO standard for their guidelines are tested before the product is HPP and after it's HPP. There are some nutrients that go down, in down arrow, some that go up, and some that stay the same or horizontal. When statistical analysis was run with limited data points here, it was concluded that there is no trend indicating nutrient destruction with HPP. The difference in analytical results between the before and after HPP is not statistically significant. So I want to, to close this presentation with a few key takeaways. HPP is not perfect, but it is the best non-thermal processing technology to make safe raw pet foods with little to no impact on nutrients. Optimizing pathogen kill requires an in-depth understanding of formulation and operational interactions. It is important to remember that everything must be built on the foundation of solid food safety best practices, coupled with valid verification processes. Because if you have only valid verification processes without a solid foundation of food safety, you might still get HPP product that come back to be positive and pathogens because there is cross-contamination coming in during the making of the product, even after HPP. So I'm taking any questions if you have any questions for me now.
I think that we can take one question. Is there? If there is, otherwise there is one, Nick. So just wait 1 second and we'll get the mic to him. And
okay.
The questions we will actually take then at the panel later on this afternoon. And Susy has actually very nicely, although all the way in Indonesia, promised to
yes
in the panel. So please, Nick.
Hi, Susy. Thank you very much for your presentation. I'm not sure if it's your field, but how many non-pathogenic bacteria do you think are harmed in high pressure processing?
When we did the validation study, actually, we enumerate the background microflora that's naturally occurring, like lactic acid and some others. We don't know how many. There must be hundreds and hundreds of different naturally occurring microflora there. But we know that even the background microflora do get affected by HPP, not all the way, but these are non-spoilage microorganisms and non-pathogenic. So we are not concerned about its presence. We just, in our one of our validation study, we just want to know how much log reduction it experienced, and we saw about 4 to 5 logs on all these background microflora, like the lactic acid bacteria.
Okay, thank you.
Actually, yes, could somebody throw the mic to Susan?
Asking whether, the thing is that the HPP is not a sterilization. HPP is pasteurization. So these foods are not sterile, and companies that use HPP are going to be testing for the pathogens. And if they're testing and they say it's zero tolerance, they don't go out with pathogen in them, but they don't go out sterile. There are still some bacteria left.
Absolutely correct, Susan. Are there any more questions?
Yes, we just 1 second. Can you throw it to Connor, because the question can't be heard by the others? And Susy, while we wait, do you think you could unshare so we can put on this next slideshow, please? Stop sharing. You just push the green button.
Yep, I just did.
Super, thanks.
Hi, Susie. Thanks for the talk. Has anybody looked at the response to the animal's biome and consuming HPP protein?
You mean doing some double-blind study, research, clinical study? Is that what you're asking?
A before and after of feeding HP, like the protein looks slightly denatured. You showed us in the photographs. The protein
Yes.
looks slightly denatured the higher you go. You go up to 6 megapascals. It was only 4 in the pictures. And so how does the dog's or human host biome respond to that protein? Does it change in response to those foods?
I don't know the answer to that question. Maybe anybody has more knowledge on that than I am. I'm not aware of anybody looking into that, but Susan, have you come across of any papers to talk about it?
Well, she said no, she hasn't. All right, cheers, Susie. Thanks very much.
Thank you.